Good morning, students. We are going to continue chapter 8 of NCERT, that is Human Health and Diseases. Up to now, we have discussed about so many diseases, especially the infectious one. So now it's time to learn more about immune system. How our immune system is being designed to kill a particular pathogen. So what actually the components are there in our immune system and how it is going to work, which we are going to discuss in this presentation. So let's begin. First of all, we need to understand the concept of immunity. Always understand the concept of immunity with the help of an analogy, that is a house which is going to be protected with the help of a guard, and one robber might come. So that robber is going to be defeated in its motive by the guard. So what that house means in the immunity, that is human body, and immune system is the guard, and antigen. Antigen are the robbers which can come to the body or the house, and they can harm the house or the body. So, how God is going to protect the house from the robbers? In case of like analogy, we can say with the help of bullets or any type of defensive system he might be having. In that case, when we compare with that of human bodies, these are actually antibodies. Okay, so antigen is the robber, which can be your any sort of pathogen, toxin, or mighty like pollens, which can cause so many immune response. So our immune system is capable to fight against all these things which are foreign to our body, which are not being required by the body, with the help of a secretion, that is antibodies. Antibodies are actually proteinaceous component, and that is going to be released by the guard or the bullets, by which it is going to protect the house from the robbers. Okay, so this concept need to be understand with the help of this thing, particularly this thing. This is very important. Now the term antigen antibody will be frequently used in this chapter. First of all, we need to understand the term antigen. Antigen are the foreign substances inside the body, example chemicals, microorganism, toxins, or any sort of pollen which is not being required by the body. It triggers immune system to produce antibodies. Antibodies are the bullets which guard or the immune system can be used to kill antigen. Okay, so what are antibodies actually? These are also referred as immunoglobulins. These are protective protein produced by the immune system in response to the antigen. Now come to the immunity. What actually immunity talks about? It is the ability of an organism to resist or defend itself from any development of a disease. We have two types of immunity, that is innate and acquired. Innate immunity is by birth immunity, and acquired is being that kind of immunity which is gradually being developed into the human body. It can be active or it can be passive. We will talk about one by one. So first of all, we are going to talk about innate immunity. That is by birth, by God, we have this kind of immunity. Okay, that is going to be inherited from the parents, of course. It is very non-specific. It can't recognize the antigen very well. So it is a general kind of immunity we have. So it is accomplished by providing different kinds of barriers like physical, physiological, cellular, and cytokine. Out of these, all these barriers are very important. But from examination point of view, cytokine barriers are the most favorable by the preferred by the CBSE. So cytokine barriers are very important. You can't afford this concept to skip down from your notes or from your learning point of view. So, kindly keep this in mind that these four barriers are very, very important from examination point of view, and you need to keep on recalling cytokine barriers again and again during your course of revision. So first of all, we are discussing about physical barrier. As the word suggests, these are just a physical kind of things we have as a mere sort of protection from any sort of pathogen, like skin, mucous membranes in the digestive or respiratory tract, and urinogenital microorganisms, which are going to provide a sort of not actual, they are not specific, that's why I'm using this word very cautiously, a sort of immunity to our body. That is by birth. We do not develop skin after the birth. It is by birth we are having that. It is a kind of physical barrier, and in case of mucous lining, you can see all of the this digestive tract is going to be protected with the help of the mucus, which is going to kill any sort of pathogen. And mucus lining is also being present in your nostril, in your trachea, and that is going to protect your body. Now come to the urinogenital microorganism. You can see a lot of microorganisms going to be surviving your urinogenital tract. And these bacteria are very, very important to uh, have a role in a sort of fighting with any foreign or dangerous substance. So that kind of barrier we are actually having with us by birth. Next is physiological barrier. It is very easy to understand. Sweat, tears in eyes, and acid in stomach, saliva. They are physiological barriers. These are kind of substance or enzymes being produced by the body to prevent any sort of microbial growth. Now come to the cellular barrier. We have a very well-fledged, fully fledged, in fact, cellular barrier. First kind of cell we used to study when we talk about immune system, that is WBCs. WBCs are going to play a vital role in building up the immunity and fighting with that of the pathogen. Out of all WBCs, PMNL, that is polymorphonuclear leukocytes, along with the neutrophils, play a vital role in killing the pathogens. Monocytes having the phagocytic capacity, they are going to lyse any sort of pathogen along with that of the macrophages. So macrophages are the big huge cells you have already seen in your class 11th, and they have the phagocytic capacity to digest any sort of foreign substance which is not being required by the body, or being detected as a non-self that does not belong to your body. It is going to be digested by PMNL, monocyte, or macrophages. They have the capacity to destroy any sort of pathogens along with the chemicals or toxins being present in the body or the bloodstream. So they play a vital role there. Now, the most important thing we have to do here is the cytokine barrier. My animation is somehow distorted. I don't know why it has been distorted, but I will explain you in a great detail about this. First of all, cytokine barriers are in terms of proteins, especially the interferons. Interferons are going to be released when one cell is going to be infected by any sort of virus. This is our cell which is going to be infected by any sort of virus. Virus just come inside of it, and virus ultimately what it do? It makes multiple copies of its genetic material and make its own particle like this. We will have multiple particles like this. So when these particles are going to be formed, some chemicals are going to be released by this infected cell. These chemicals are referred as what? Interferons. These are actually being released in case of viral infection, so they are very, very particular. Interferons. Okay, these are interferons being released, And these interferons, what they are ultimately going to do, they are going to send a message to the uninfected cell that protect yourself from any sort of viral infection. So, how they are going to commute? They are just going to be released out of the cells. They just reach to the cells, and we have studied, we have so many types of proteins or glycolipids being protruding out of the cell membrane. So, these protruding protein along with the glycolipids play a vital role in accepting the interferons, and they send a signal inside of the cell protect uh, yourself from any sort of viral infection. So the next time the virus which going to come and infect this cell which is already being prepared, this cell will not accept the genome of the virus and it to prevent further infection. So this is very very crucial when we are going to learn about cytokine barriers. Cytokine barriers are actually done with the help of the proteins that these proteins are interferons and they are specific in case of viral infection. What we have to learn that in case of viral infection interferons are going to be released out. Okay, so this kind of learning we need to develop during the course of this chapter. By now, we have learned that innate immunity is provided by a kind of barrier that is physical, physiological, cellular, and cytokine. These four kinds of barriers we have discussed. Now it's time to learn about the immune response. How our body is going to prepare or fight with the antigens. So we have two types of cells in our blood streams. These are B lymphocytes and T lymphocytes. B lymphocytes and T lymphocytes. B lymphocytes in human are going to be produced by bone marrow. So sometimes bone marrow is correlate with that of the B lymphocyte, but this is not the case. First of all, you need to understand when B lymphocytes were firstly identified and Aves, they were extracted from bursa of fabricus. Bursa of fabricus in birds or Aves, you can say simply. I'm writing birds because BB will be common in all of the cases you can easily recall. So bursa of fabricus is responsible for the production of the B lymphocytes from where the word B actually comes. Because most of the teacher get confused from where the B lymphocytes, B, the word has come or arrived from. That is bursa of fabricus in birds. In human, of course, it is co-relevant with the bone marrow because bone marrow is the actual site of production of B lymphocytes. They are the cells which are responsible for releasing antibodies or the bullets against the robbers or against the antigen. So these cells are vital for our immune system. Very, very important cells. They can fight with any of the antigen, but they are very antigen specific. Okay, so if there is a particular kind of antigen, a particular kind of antibody is going to be released by the B lymphocytes. Okay, we have again two types of lymphocytes, the B lymphocytes, that is plasma and mammary. Plasma lymphocytes or plasma B lymphocytes or plasma B cells, they are actually going to produce the antibody and destroy the antigen. But memory B lymphocytes are very very important because they are specific to the antigen and they are long lived and they are important for the secondary encounter. What is secondary encounter, secondary response, what is primary response? First of all, imagine a body. I'm making a raw body like this, and first time a pathogen just come inside the body. So body is not able to recognize all of a sudden. It takes some time, and B lymphocytes get activated and release antibodies. This type of response is called primary response because body is first time facing uh, an immunological response against a particular kind of antigen, and might be the body is able to fight with the pathogen or toxin or able to eliminate it, but there is no surety for the same as well, because if the case the antigen is quite harmful for the body, the person can die also. So it is a hit and trial method. Body is trying to gather all sort of information and put all efforts to fight against that of the antigen. So we are considering that case when body is able to fight fully against with the antigen and able to eliminate it altogether from the body. Now come to the, this is referred as primary response. This is referred as what? Primary response. Now come to the another response when the body is being infected with the same antigen again. Now our body is quite shrewd in that case because it has already developed antibodies for the particular antigen. So it has memory also to recall when I have this kind of interaction. Just suppose that first time primary response happened at 0 age, and the second infection just happened after a lapse of 25 years. It is able to recall that we have that kind of infection. So second encounter is referred as secondary response. Now body is full-fledged, you have memory, how I am capable to fight that particular kind of antigen. So these memory B cells are going to help to create a memory for the lasting impact. Now come to the T lymphocyte. They are also referred as helper T cells or TH helper T cells. I'm just writing the abbreviation of the same. They don't produce any sort of antibodies, but they make and they stimulate B cells to secrete a particular kind of antibody against a particular antigen. How it is going to help? First of all, it take message from the infected cell and transmit that message to the plasma cells that please secrete that kind of antibody. And it also stimulate memory T cells to create a memory simultaneously for the same kind of antigen. So the helper T cells or the T cells, T helper cells, TH cells, they are not going to produce any sort of antibody, but they help or stimulate B cells to produce a particular kind of antibody. This is very, very important point. Now, the another type of immunity we have to discuss beside innate immunity, that is acquired immunity. Acquired immunity, as the word suggests, is not going to be present by default in the body. It is being acquired just after the birth. Okay, it is pathogen specific and it is able to distinguish diverse type of foreign antigens or molecules. When the immune system encounters a pathogen for the first time, a primary response is going to be developed, and a second time, the secondary or amnestic response is going to be developed. So, first encounter when we have discussed for the very first time, antigen is going to infect the body. and that response is referred as primary response. And if the memory has been developed, we have that kind of nervous system, immune system with us. And if we have memory, we can develop secondary or amnestic response in the same case. So that is because of the memory B cells. Okay. So I hope this part is quite clear to you. Now come to the two types of acquired immunity, that is humoral and cell mediated. Humoral, as the word suggests, humoral. You have discussed in the case of eye, we have humoral chamber, vitreous chamber. So humoral is just referred to as a body fluid, and it is mediated with the antibodies that is going to be secreted by the B cells and transferred by the blood and the lymph. Cell mediated is generally with the help of the cells, that is T lymphocyte. And T lymphocytes are responsible for the activation of the B cells, either plasma type or the memory type. So, cell mediated kind of immunity just happen on the basis of recognition on the capacity, that is self and non-self. It is very very important in case of graft rejection. We can't take heart or any sort of tissue from any body. We have the capacity of disjunction between self and non-self. That type of response is generally mediated with the help of cells, that is T lymphocytes. Okay. In graft rejection case, we have to use immunosuppressant. Why we have to use? Because we want to suppress our own immunity. We don't want our immunity to work on the graft we have placed. Okay, inside the human body. Now we have two types of immunity again on the basis of antibody production or ready-made antibodies we can also inject. First of all, active immunity is that kind of immunity which is going to be developed when a pathogen, dead or living, is going to incubate on our body, and our body is going to secrete lots of antibodies. That is active immunity. Active immunity can be natural or artificial also. Why natural or artificial? Natural or artificial. My handwriting by the annotation is very bad. Natural and artificial. What is natural active immunity? Natural active immunity is that kind of immunity which is being developed by the natural response we have. Okay? When a pathogen interacts in our body, antibodies are going to be secreted. This is a default system of our body. Artificial, when we inject forcefully into the body some sort of inactive or less active or dead form into the body, and that kind of response being generated like a production of antibodies, that is referred as artificial. It has no or only a few side effects. It is slow but long lasting, as you know. It is going to develop memory cells as well. It takes time to get its response. Example, vaccination for polio or hepatitis A. Injecting microbes deliberately during immunization. That's what I'm talking about. Artificial. Deliberately, we are going to inject with the help of vaccine. That also create active immunity. Now, what is passive immunity? Passive immunity is that kind of immunity when we just take antibodies, uh, which are quite ready-made, and that going to be injected into the person's body directly. It may cause a lots of reaction. It is very fast but lasts for a few days only, because it not led to the development of the memory. It is very fast, like administration of tetanus antitoxin, or in case of rabies also. And nowadays, for COVID-19, we have developed plasma therapy on this passive immunity because we are injecting the ready-made antibodies in the same. Fetus receive some antibodies directly from that of the mother, and that is also a passive immunity because fetus is not going to take not going to take the chance to develop immunity by itself. Some of the immunity is going to be provided by the mother's milk and placenta. So that type of immunity is referred as what passive immunity. Now come to the structure, most wonderful structure, that is of antibodies. Antibodies are also referred as immunoglobulins. We have four kinds of immunoglobulin: IgA, M, E, and G. They are actually Y-shaped structure. We have Y-shaped structure. Each antibody comprises of four polypeptide. You can see two long chain and two smaller chains. So heavy and light chain. And you can easily recognize sulfur bonds. Beautiful sulfur bonds are there. They are going to form peptide bond between two of the amino acid. If this is one amino acid and this is another amino acid, a peptide bond is going to be formed with the help of sulfur bond. So you can see a beautiful, beautiful series of sulfur bonds over here. And this zigzag pattern is here. Zigzag pattern is a antigen binding site, any sort of foreign pathogen, chemicals, anything that will come and that will bind here only. So IgE is the most prevalent antibody in human system, and IgA and IgM are somewhat lesser than that of the IgG. IgG, that is immunoglobulin globulus kind of protein, is going to be present. IgA is going to be provided by the cholesterol. I just want to discuss a little bit about the structure. It is a Y-shaped structure. The normal structure is Y-shaped. But in case of IgA, we have a dimeric form. That is, here is one Y and we have another Y over here. Okay, this kind of a structure. Dimer structure can also be there. In case of IgM, we have a pentagonal structure. This is pentagonal, and this is five immunoglobulins being present over there. Okay, and these are IgE and IgG are the monomeric. So we can have different type of immunoglobulin on the basis of Y, that is antibody, typical antibody structure. Now moving to the next part, vaccination and immunization. Vaccination is that kind of process when we provide inactive or dead form or weakened form of any sort of pathogen or protein against a particular kind of disease. Immunization is a process by which body is going to develop antibodies against the vaccine, that is primary response, and it is going to protect uh, the body for the further encounter or the secondary response. Like in case of hepatitis A, if the person has already been vaccinized for hepatitis A, already it has developed a primary response, which is a sort of passive immunity or active immunity. It is active, right? Active immunity we are going to done with the artificial method, and the body is going to release lots of antibodies for the same, and ultimately it's going to create the memory. If further infection going to happen in the body, it will cause a secondary response. So secondary response will be the faster one. Vaccination provides immunization after gap. Okay, so there is a time lag between vaccination and immunization. Vaccination are and immunization are based on the property of memory. That is very very integral point. That is based on the memory. Okay. Now moving to the next part. Vaccine generates a memory B and T cells that recognize the pathogen in the further or subsequent infection. If a quick immune response is needed, as in case of tetanus or a snake bite, we can use antitoxin or ready-made antibodies from the patient, that is referred as passive immunization. By recombinant DNA technologies, we can produce vaccines on the large scale, like that of the hepatitis B we have produced from that of the yeast, that is yak, we have already discussed in chapter 6. Now, the most interesting phenomena, that is allergy. Allergy is that kind of immune response which is quite exaggerated. Okay, hypersensitive reaction to the immune system against a particular kind of antigen. The subsequent agent which produce an immune response in an individual is called allergens. Allergens can be of any type, like pollens, chemicals, dust. These kind of allergens can be there. IgE is going to be produced in that case, like and the chemicals which are going to cause allergy. These are histamine and serotonin, which are ultimately released from that of the mast cells. Symptoms of the allergy are sneezing, watery eyes, running nose, difficulty in breathing. So, what are the cure? We can take antihistamine, adrenaline, or steroid for the same. To determine cause of the allergy, patient is injected with a small dose of suspected allergens, and the reactions are going to be studied. Modern day lifestyle has lowered immunity and increased our sensitivity, that is hypersensitivity towards allergens. Okay, so I hope that part is quite clear to each and every child. You can recommend this video again and again, so you have an holistic understanding about the concept. That's all for today. Thank you, students.